Good evening, everybody. Can you hear me okay? So my name's Saif. Thanks so much for inviting me today, and thank you for the CSAR Award Committee for awarding me a prize last year. So my talk will be about my PhD, the 4 years that I've done, and these four sort of random pictures bring together the focus of my research. So the picture on the right-hand side is a cycle lane, actually, in the south of Cambridge, near Great Shelford, and on that they've got the full genetic code of the BRCA2 gene along this whole cycle path. And actually on the left-hand side, there are three celebrities who have been affected in some way, so Angelina Jolie, Pierce Brosnan, and Christina Applegate, by either having a mutation in the BRCA gene or having family members who have been affected by cancer as a result. And so what my research is focused on is trying to understand in more detail why mutations in the BRCA2 gene lead to cancer. And I'll be talking a little bit about that over the next 5 minutes or so with you. So why is it an important question to be addressing? Well, around 10 million people across the world will carry inherited mutations in BRCA2, which could lead to cancer. And that's around the population of London. And so there's actually multiple types of cancer that these individuals are predisposed to. But we don't yet fully understand why this happens. And so to illustrate this, if we look at women at risk of breast cancer, so by the age of 70, if you have a BRCA mutation, so you can have either BRCA1 or BRCA2 gene, mutations, about half of women will go on to get cancer by the age of 70, whereas about only 12% of the normal population would. But it's not just limited to breast cancer, so prostate cancer, ovarian cancer, and pancreatic cancer. All these cancers are linked to this mutation. And what's important to take away from this slide is that we might as well toss a coin when we see someone in clinic who's got a mutation, because it's only a 50% chance they'll actually get cancer. So if we're talking to them about having surgery to remove their breasts or ovaries to reduce the risk of cancer, it's only in about 50% of women that would actually have made a difference. So to understand why BRCA2 might be leading to cancer, we have to delve into the biology. And just to illustrate that, if we take someone and just zoomed in on their pancreas, then we would see that our pancreas is made up of cells that are organized into tissue, so that's a gland. In more detail, then we have a cell with a nucleus, so those sort of X-shaped things are the chromosomes, which carry our genetic code, which is encoded in DNA. And actually, our DNA is at risk of being damaged every single second of every single day. That's due to reactions that are happening within our cells all the time, making byproducts that can damage the DNA, or just through the environment, through radiation or tobacco smoke, for instance. And what we know, and a lot of the work was done in Cambridge decades ago, was that the BRCA2 gene helps to repair damage to DNA, and therefore in that way is linked to reducing the risk of cancer. But actually in recent years we've also understood that BRCA2 works in lots of different ways. And what my research is focused on is trying to understand how it works at this sort of pre-division checkpoint. I'll talk about what that is briefly here. So our cells are dividing generally, but at the time of division they must make sure that the genetic code on them is pretty much 100% accurate. If that's not the case, then these cells might die, or they might carry mutations into the daughter cells, which could lead to cancer in future. And to illustrate that, here's a video of a cell dividing with damaged DNA. If you look at the one in the bottom right, it's trying to divide, but because the DNA is damaged, then both cells die, basically. So it's obviously not something that you want to be happening. So as I said, BRCA2 has recently been implicated in this pre-division checkpoint to make sure that when our cells divide, they're carrying healthy DNA. So that was the focus of my research project. And to distill about 4 years of work into the next minute or so, I'll just say that my research has helped to identify a protein, actually a couple of proteins, that work together with BRCA2 to help function at this checkpoint. And to illustrate that with one of the results that, once I've done so, what could the impact of that be, is that I've looked to see that actually if we how that can affect even when we give drugs to cancer. So just to briefly explain this slide, the top line, the blue line, is just normal cells if they were given a chemotherapy drug and what percentage of them are still alive. If I had, say, a drug against one of the proteins that I identified, then it wouldn't really make much difference. But if a cell had a mutation in BRCA2, then you can see that at the higher concentrations, more of these cells are dying. But what's been quite interesting to find is that if I say if it was a BRCA2 mutant cell which had a mutation, and I then inhibited the protein, say via a drug, then that seems to have a really effective cell killing effect in this situation. So what are the applications of the research that I am doing? So it could have an implication for the cancer screening as well as counseling, because as I mentioned earlier, we might be able to know where these mutations are, but we don't know what the impacts are. And so what my research has done is shown where on the BRCA2 protein my proteins interact. So if we know there's a mutation in that area, then that might be more clinically significant. Also, if we understand better about how BRCA2 is working to prevent cancer, then potentially we could design strategies to prevent people getting cancer in the first place. But if they go on to develop cancer, there's a big focus now towards personalizing medicine. So we use the bespoke treatment in an individual cancer. And actually, what my data has shown, that if we potentially target some of the proteins that I've identified in BRCA2 mutation carriers cancers, then that might be effective. And in terms of my sort of future work, I'm looking to hopefully do projects with AstraZeneca, who've moved to Cambridge, to get a better handle on sort of drug discovery programs in the context of these proteins that repair our DNA. So at that point, I just would like to say thank you to everybody in the audience listening to my talk, but my supervisor, who's been very supportive through my time during my PhD, and also, of course, the CSAR Award Committee, who've been who selected my project, and I think most importantly, everybody who helps donate to the CSAR and make this possible. It means that I'll be able to take part in a conference in the US hopefully next year. So thank you very much.